Charla que va a presentar el doctor Reiden van den Horn. Les voy a leer una pequeña biografía de su antes de darle la palabra. El doctor Reiner van den Horn estudió química en la Universidad de Leiden, Bélgica, centrándose en la biología molecular y la bioquímica de las plantas. Completó su doctorado en 2001 en fitopatología molecular en la Universidad de Wageningen, Países Bajos, disculpen la pronunciación, con el doctor Pierre de Ville, donde trabajó en las proteínas de resistencia CF del tomate. Obtuvo una beca Beni del Consejo de Investigación holandés y comenzó su propia investigación introduciendo el perfil de proteínas basado en la actividad ADPP en plantas, que estableció como científico visitante en el laboratorio Sainsbury en Norwich, Reino Unido. Inauguró su propio laboratorio en 2005 en el Max Planck Institute en Colonia, Alemania, como parte del Centro de Genómica de la Sociedad Max Planck. Posteriormente, obtuvo una beca RC Consolidator y se trasladó a la Universidad de Oxford en 2013 como profesor asociado y tutor de ciencias vegetales en Somerville College. Se convirtió en profesor titular en 2017 y profesor de investigación RS4 en 2022, cuando obtuvo una beca RC Advance. Su investigación se centra en mejorar la expresión transitoria de proteínas en plantas y en utilizar la proteinómica química y el modelado estructural para descubrir los mecanismos de manipulación extracelular del huésped empleados por los microbios al colonizar el aroplasto. Y bueno, esperamos que disfruten de esta conferencia. La idea es que los estudiantes charlen un poco con el investigador, bastante renombre. Agradecemos a la doctora Claudia por la oportunidad de traer a tan importante persona y que tengan la posibilidad de platicar con ellos. Doctor, please, all yours. Thank you.
Okay, thanks for the introduction. My Spanish is not that good, but I picked up some words and I think it's all correct. Thanks for being here. I will talk about plant pathogens interactions today. So this is not my office. This is Oxford. This is the library in the center of Oxford, and the labs are a kilometer a bit down the road. So my lab is investigating the interaction between plants and pathogens. And what you see here are 3 infections of tomato leaves with 3 different pathogens: a bacterium, a mycete, and a fungus. What they all have in common is that they colonize the apoplast, the extracellular space within a leaf. And it's a very interesting environment because you have changing light, temperature, humidity, but it's also a living surface. The plant responds to infection, and it does so by secreting toxic molecules, and that includes hydrolytic enzymes that we think have an impact on the pathogen. But of course, these pathogens have adapted. They cause disease. So we think they're able to suppress the most important hydrolases by secreting inhibitors that target these hydrolases. So the mission of the lab is to identify which secreted hydrolases are inhibited, and I ask 2 questions. Why is it inhibited, and how is it inhibited? The idea is that with this we can discover novel extracellular components of immunity that we can then later engineer such that they are insensitive for manipulation, and thereby we reduce the susceptibility of the plant. Now, to do that research, we take advantage of a model system. So we have a model plant interacting with a model pathogen. So that is the interaction between the Pagana pentamina plant and Pseudomonas syringae. This is pathovark tomato DC3000. This is the most investigated plant pathogen in the world. So we know a lot about this. We actually use the HOP-Q mutant because that mutant is able to cause the speck disease on the plant. The wild type strain cannot, but the HOP-Q mutant can cause disease. So that's the infection that we investigate. And of course, because this is DC3000, we know a lot about how they manipulate the host. mostly through the injection of these type 3 effectors inside the plant cell through a type 3 secretion system. What we don't know about much is what happens in the extracellular space, even though this is a plant pathogen, the model plant pathogen. So the approaches that we're taking is, one hand, chemical proteomics, off-fold multimer, and reverse genetics. And today I will give you examples of each of those approaches. Okay, so first of all, how do we get access to how are we able to study the apiplast? That is by isolating apiplastic fluids from infected plants. How do we do that? We submerge leaves in water. We put some stones on it to really make sure that they float. Then we apply the vacuum, so we pull out the air, and then we release vacuum, and then the water gets inside the leaf. We then take the leaves out, dry them on the surface, roll them up, and then transfer them in this cylinder, which we then centrifugate at low speed. So we don't break the leaves, but we spin out all the apoplastic fluids, which we collect in a tube underneath. That is for us a very important sample to work with, and that is for us the gold mine that we need to investigate. So what we have in here are two proteomes, one from the plant and one from the pathogen, and they interact with each other. And what we're mostly interested in is what is the outcome of this interaction with respect to enzyme activity, because that dictates how these plants and pathogens interact. Now, this functional proteomics layer of information can be displayed using activity-based protein profiling. What's my screen? Not sure. Seems to be an issue with the computer here. Okay, I don't know what happened. And it seems to restart again. I don't know why. Okay. 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 I will take you where I was. Yeah. So I was explaining that we use activity-based protein profiling. So that is a technique that we use to detect which proteins are active in a proteome. And it's based on labeling of the proteome with a small molecule probe, which mimics the substrate but locks the enzymatic mechanism in the covalent intermediate state. So that allows you then to monitor which enzymes are active. You do not need to purify the proteins, you do not need to know the substrates, and you can display multiple protein activities simultaneously in crude extracts and also in living organisms. Just an example of that is this probe, JJB70. It's a probe for glycosidases. And this is a probe that Pala Kumaran, a PhD student in my lab, started working with. This is a probe that we got from the Overkleef lab in Leiden, and it has a fluorescent group over here and a reactive group over here. This reactive group will react with the active site glutamates of glycosidases. So when you incubate this probe with an apoplastic fluids from Nicotiana benthamiana and separate the proteins on a protein gel and scan for fluorescence, you can see a number of proteins that are labeled. These proteins were active when we did the labeling. We know by mass spectrometry that these are glycosidases, and here we have a galactosidase. You can see that the signals are much more reduced when you take samples from infected plants, especially this signal is BGAL1, is strongly suppressed. Now, if you mix the labeled samples, then of course you get the exact average of those signals. You can quantify that and plot it in a graph. But if you first mix the sample and then do the labeling, you actually have less labeling. So that means there's something in the apoplast of infected plants that is able to suppress the labeling of the enzymes from the non-infected plants. So that indicates this inhibition. This method is called convolution ABP. So this raises the two questions. This inhibition, why is it inhibited and how is it inhibited? And I will address both questions today. So first, does BGAL act in immunity? So we removed B-gall by genome editing from the plants. The mutant plants look absolutely normal. But when you isolate apoplastic fluids from those plants, you can see they have a drastically reduced beta-galactosidase activity. And that is done with a fluorophore that has 2 galactose on it, so it becomes fluorescent when you remove the galactose. So B-gall is actually the major beta-galactosidase activity in the apoplast. But we don't see a phenotype. However, when we do infection assays, we do see that the mutant plants are more susceptible, not a lot, but significantly. So the plants are more susceptible, and that means that B-gall does have a role in immunity, and that explains why it is suppressed during infection. So the next question is, of course, how does it do that? How does B-gall 1 achieve this suppression? Initially, we thought, okay, maybe it acts on the bacteria itself, but we could not establish good assays to show that. But then we speculated maybe B-gall actually releases something from the bacteria that is recognized by the plants. And typically this is associated with the release of reactive oxygen species called the oxidative burst. So we set up an assay to detect that type of infections, or the release of the elicitor. So what we did is we mixed Pseudomonas syringae with apoplastic fluids containing B-gall or not, and then added that to a leaf disc floating on luminol HRP. If the leaf disc recognizes an elicitor, then they will release reactive oxygen species that will light up the luminol, and we can see that in a plate reader. So we can detect the oxidative burst. Now, this is indeed what works. You see a very nice oxidative burst peaking about 25 minutes after adding this mixture to the leaf disc. But it is dependent on B-gall, indeed. So without B-gall, we don't get that release. So something is released from the bacteria by B-gall, and that is recognized by leaf discs of Nicotiana benthamyrana. We also, but if you take leaf discs of Arabidopsis thaliana, you also can detect this. So that indicates that what is recognized by both, by different plant species. So it must be quite a conserved perception mechanism. Now, because this is a response in Arabidopsis, we could take advantage of lots of mutants, because we started thinking what we need to look for now is a receptor that detects a glycan. Of course, B-gall is a glycosidase. So we need to have a receptor for glycan. So we tested different mutants, but never found something that was insensitive for this elicitor. But then we also included FLS2. So FLS2 mutants are completely blind for this inhibitor, elicitor. So that means that FLS2 is receiving this elicitor. Now, FLS2 is the flagellin receptor. It's a very famous receptor, very well studied. So if that's true, then we expect that what is released here must be flagellin. And indeed, when we used a fly C mutant, which doesn't make flagellin, the elicitor is no longer released. So somehow B-gall 1 releases or mediates the release of the flagellin elicitor. And this is confusing because flagellin is a protein and B-gall is a glycosidase. So how could a glycosidase be involved in the release of a protein-based elicitor? Well, it's actually explainable because the flagellin peptide that is recognized is 22 amino acids long and is deeply buried into the polymer. So this is the monomer, but this is a polymer of flagellin. And that polymer is decorated with a glycan. Every monomer has 6 O-glycans. And the glycan that we have here is on the serine residue and contains 2 or 3 rhamnoses and a modified viosamine. And we know this because of really good work from Yuki Ichinose at Yokohama University, who described this glycan, not only for the strain that we use, but also for a different strain that was the PTA6605. That is a Apathovar tabaci. And the reason why he studied this one is because it swims better. It has more flagella, and therefore research on the glycosylation of flagella was easier in this strain. Now, luckily, this strain is also pathogenic on bentamiana, but it makes no BGAL1 inhibitor, which means our phenotypes are a bit stronger. So we decided to then test this, if this strain behaves the same way. So we take this strain, incubate it with the apoplastic fluids containing BGAL0, and then add it to a leaf disc. And that leaf disc responds very clearly more in the presence of BGAL than in the absence of BGAL. So also here, the elicitor is released by BGAL from those bacteria. And also here, BGAL mutants are more susceptible. So that means it does act in immunity also on this particular strain. So now we could take advantage of the mutants that were described before. So this is a mutant that still makes flagellin, but it is not glycosylated. If we incubate bacteria, these bacteria with apoplastic fluids, we release the elicitor very quickly in the absence or presence of BGAL. It doesn't matter, and it's a very strong response. So clearly, the glycan protects against the release of the flagellin elicitor. Now, if you do infection assays, you can see that in wild-type plants, they don't grow very well compared to the wild-type bacteria, but that is because recognition is switched on instantly. But more importantly, is that the BGAL mutants are not more susceptible. So BGAL only acts in immunity when the bacteria carry a glycan on the flagellin. We could even zoom in a bit more because there were mutants described that still make rhamnose glycans but lack the modified viosamine. And they behave very similar because they are also triggering the immune response instantly, irrespective of BGAL1, and they don't grow very well, and BGAL mutants don't increase the susceptibility. So that means here, again, BGAL only acts in immunity when the strains carry a modified viosamine. And that makes a lot of sense because the modified viosamine has structural similarity to galactose, which is the described substrate for BGAL. We also have another strain, because not all the strains have the same glycans. So this is a Pathovar syringae strain, which has most likely a glutec at the terminal glycan. And that strain is not recognized. And it grows very well on Nicotiana bentamiana, and it is not affected by the presence or absence of BGAL itself. So what we have here is basically the very first step in most likely a proteolytic cascade that results in the release of the flagellin elicitor that is perceived by the flagellin receptor. And we think that we speculate that proteases probably are involved in this step. So this is with the 6605 strain, and to mitigate, to suppress this recognition, TC1000 is able to make the BGAL inhibitor, and a different strain makes a glycan that is not sensitive for BGAL. So you see very nice an arms race between the plant and the pathogen trying to avoid recognition. Now, I want to say a little bit about the protease work, because we did look into this much more in detail. And I just give you a summary slide because you can read the rest. But basically what we thought would happen is that after the polymer is monomerized, and we think BGAL actually helps in the monomerization of this process, the flagellin peptide should be released from the monomer. And we were looking for the protease that is responsible for that, and actually we did identify that. It's a subtilase 5.2, SPT 5.2. But the problem that we noticed is that actually that same subtilase also cleaves within the peptide. So, and it does that in the precursor, but also in the peptide itself. So actually SPT 5.2 inactivates flagellin, and it's very similar to work that was published also recently, where they made similar observations in Arabidopsis. And the way to interpret that is actually the bacteria are probably taking advantage of the host. They take advantage of the protease that the plant has to avoid recognition. They make sure that the fragments that are recognized by the plants are actually unstable, and because they're degraded by plant proteases. Okay, so that was very short, because if you want to read more, you can look it up. And I wanted to talk a lot more about the inhibitor because we have a really exciting, if we have the inhibitor, we know what the inhibitor is, and it's something new. And this is a project that we started working on 10 years ago, pioneering work by Bala again, and most of the work was done by T, who now, actually both of them, he has a lab in India, and T just started a lab in Bangkok. And we collaborate with a lot of other people for good reasons. So, and actually, this is T, visited him in December, showing the molecule here in his hand. So I will show you. So the molecule, the BGAL inhibitor, we call glycosirin, because it's not only inhibiting the BGAL, but also other enzymes. So it's a very general inhibitor. So this was the question, right? So how is that BGAL enzyme suppressed during infection? So we could already describe a bit of properties of this inhibitor. We know it is small, it's heat stable, it's non-volatile, it's polar and basic, and it is secreted by the bacteria when grown on minimal media. So now we did a trick to identify the cluster, the genes that are required for biosynthesis of this inhibitor. We took advantage of Laxet, and you all know Laxet, I think, if you have done some molecular biology, because Laxet is an enzyme from E. coli that encodes for beta-galactosidase. And you can visualize the production of this galactosidase if you put X-gal in the plate. So that's a substrate that turns these colonies blue. The blue ones make the enzyme, and the white ones cannot make the enzymes, and that's why they are white. So okay, that's handy. So let's try if we could use that. Let's put it into DC3000, and indeed, if we you get blue colonies if you grow them on rich medium, but on minimal medium, they're white. They're white because the inhibitor is produced. So now we could use it, do a screen. We could mutagenize the genome with a TN5 transposon and then screen for mutants that are blue. So we picked up 300 blue colonies and started sequencing the insertion site of the transposon. And in this way, we could identify 10 mutants in a biosynthetic gene that consists of biosynthetic gene cluster that consists of 5 genes, a dehydrogenase, a reductase, a phosphatase, something of unknown function, and a transporter. We have knocked out individual genes and learned that A and B are essential, C and E are partially required, and B, we don't know. It has no effect if we remove it on inhibitor production. Now, besides the 10 mutants, we had, of course, many more mutants, and it appears that those, oh, forgot this part. So, to be sure that this was the inhibitor, we knocked out the entire cluster, and we did a Laxet inhibition assay. So we take the enzyme and mix it with the supernatant of a bacterial culture. If there's an inhibitor, the enzyme is inactivated, and we would measure that. So here we can see that wild-type bacteria produce an inhibitor because the activity is severely suppressed, and the mutant that lacks the entire cluster is unable to do so. Now that mutant is also less virulent in plant growth assays. So if you put it on a plant, the mutant grows less fast. So it is actually indeed a component that is required for virulence. We also put back the cluster in the mutant. And of course, you get inhibitor production back. But also, if you do that in E. coli, then E. coli starts making the inhibitor. If you give the cluster to E. coli, that cluster is sufficient to make the inhibitor in E. coli. So we have that. But we also had many other mutations. And those mutations where we sequence the insertion sites, we noticed that they are actually in the regulatory pathway that is needed to switch on the biosynthetic cluster. So we have here, and that is explained by the fact that the cluster contains a HARP box, which is activated by HARP-L. and we found 12 mutants in HARP-L. HARP-L is activated by HARP-RS, and we found 32 mutants there. And HARP-RS is activated by HPS, and we found 15 mutants in there. This valence pathway was actually already known because it regulates the type 3 secretion system and type 3 effectors, including AVR-PTOB. And to confirm this, we did RT-PCR on strains growing in minimal medium to detect the expression of this cluster by looking at GSNA in the different mutants. And you can see that in these mutants, this cluster is no longer activated. So that indicates that this cluster is controlled by valence regulation. It's only switched on during infection. And indeed, when we hooked up the promoter to a luciferase overreading frame, we could indeed demonstrate that this gene during infection is only switched on, but very early during infection, and very similar to the type 3 effector AVR-PTOB. Okay, so we know what the cluster is. This cluster actually occurs more often in plants and in Pseudomonas, very low resolution. And when we looked at the first gene, GSNA, the very first gene in the cluster, this is the dehydrogenase. That dehydrogenase also occurs in other clusters, biosynthetic clusters. And for some of those bacteria, it was known that they are able to produce immunosugars. Immunosugars are very good inhibitors of glycosidases. And the difference is that they have a nitrogen instead of an oxygen in the ring. So we thought, oh, this is an indication that the inhibitor is an immunosugar. But it's likely a bit different than the other immunosugars because our gene cluster has a RIP-D homolog called TSNB. So that's an indication that the pathway to make this immunosugar is maybe different from the classic, the so far described ones. So we wanted to know what the structure then really is. How does that structure look like? So we took again advantage of Laxet. So now we fused it to an His tag and produced the proteins in large quantities, immobilized it on a nickel NTA column, and then we saturated the enzyme with the inhibitor by passing through a lot of these supernatants of bacterial cultures until we had saturated it. And you can see that because the aluvate starts to get the inhibitor, so we know we have saturated it. We then released it and found that the released Laxet is indeed completely inactive. And then we collaborated with a group that is really good at cryo-EM. And they resolved the structure at a 1.9 angstrom resolution. That's an atomic resolution. So we can see what is the inhibitor. And if you zoom in there, you can actually see the inhibitor. It's a tiny inhibitor, but it's there. It's a 5-membered ring. You can see that, 5-membered ring. It has 2 side groups sticking upwards here. These are most likely hydroxyl groups. And there's another side group that is branched that sticks to the back. So we thought initially that that is basically this inhibitor. This is also inspired by what we knew at that stage from the biosynthesis. So we thought, okay, that is then the 5-membered ring. You have 2 hydroxyl groups, and then there's here a hydroxyl group that probably flips around and causes the electron densities on both sides. But when chemists made that, so this was done by Marcus Keizer in Essen and his students, Werner Tabak. So when they made that, they came back to us and said, well, we made it, but it's not stable. As soon as we put it in water, it rearranges, and we realized that actually that double bonds can shift to this position, and then you have an aldehyde, and that can be hydrated with a water molecule. Now, this is not normally a stable molecule, but apparently in this conformation, hydrates is stable. And interestingly, that's what we see in the structure, because that explains that branched conformation. So just to be sure, we also redid the cryo-EM now at 1.4 angstrom resolution, and we see exactly the same conformation of the inhibitor in the active site. We also measured inhibition, did inhibition assays, found that they are strong inhibitors in the nanomolar range. So this is indeed the inhibitor. Now, looking at structure, you can also see why it's such a good inhibitor. So you see here how the nitrogen in blue is able to interact with this acidic residue, it's the catalytic residue. So that is the reason. We can also see how it compares to galactose. Galactose is in light blue. And you can see that it's quite remarkable that a five-membered, by having that five-membered ring, having that dihydride, it's actually able to mimic a six-membered carbohydrate. So it's a perfect mimicry of a monosaccharide. Okay, so we know how it inhibits. We know how it looks like and why it's such a good inhibitor. Next, of course, how is it produced? I want to keep this a bit short. But the first clue was GSNB. That was the unique gene that this cluster has compared to other clusters that produce immunosugars. And we realized this is the RIPD-like reductase. So we started looking up what RIPD means, and that ends up to be involved in the riboflavin biosynthesis pathway. And if you look at the reaction that it catalyzes, we realized that that is probably what it does. We need to look for a molecule that looks like this. So it has five different carbons. It has a nitrogen, but of course that nitrogen should become in the ring and it should be dephosphorylated. Yeah. So this molecule is known. It's a known precursor called PRAN that is involved in the production of purines. And it is made by PERF and it is converted by PERD. So to confirm that this is indeed the pathway that leads to inhibitor production, we made PERF and PERD mutants. Of course, they do not grow on without adding purines to the medium. So then they can grow, and then we can show that actually, indeed, the PERF mutant is no longer able to make the inhibitor. And initially, surprisingly, it was we see that the PERD mutant is even able to make more inhibitor than the wild type can. But that makes sense because you increase the metabolic flux by avoiding the production of purines. So you have more precursor for the inhibitor production. Okay, so this is a new pathway because it is a novel immunosugar starting pathway. And what we see is if GSNB acts on this substrate, it's very likely opening that ring. What we then expect to happen is that we need to remove the phosphate. That could be done by GSNC because it is a phosphatase. And what we then need to do is to close this ring, and you need to do that by activating this hydroxyl group with the dehydrogenase. The dehydrogenase makes a ketone, and ketones are very reactive. So they would react with the amino group here, and that would result in this glycoserin, which chemically converts itself in water into the dihydrate. Now, to demonstrate that, I keep it really short, is what we did is we made those four enzymes, these A, B, and C enzyme, but also PERF. We could buy this, PRPP, and of course we added all the cofactors that are required, and then we could actually make the inhibitor with purified enzymes in vitro, so without anything else. And by inactivating the enzymatic steps at different stages using heat inactivation, we could even demonstrate that the intermediates would accumulate and that it is in this order through which this inhibitor is being made. Okay, so we know how it is made, we know what it is, and it is an important component to suppress immunity. This happens by inhibiting BGAL1. Because if this molecule is made, BGAL1 is not active, and flagellin, elicitor, is not released from the polymer. We also noticed when we did more experiments that besides this main activity, we also see an accumulation of galactosyl glycerol and trehalose. So these are disaccharides or saccharide-derived molecules that accumulate during infection only when the bacteria produce glycosyrine. So not with the mutant glycosyrine. We don't know why they accumulate, but both of them are actually osmolytes, so they can help the bacteria to survive in the apoplast by having more water around them. We also notice that the protein glycosylation of the plant is changed during infection, and also that is dependent on glycosyrine. And that makes also sense because enzymes that are involved in the reshaping of N-glycans and O-glycans are inhibited by this inhibitor. Because it does not only inhibit BGAL1, it also inhibits other beta-galactosidases, and it inhibits alpha and beta glucosidases. They tested other glycosidases, they are not inhibited, but it is in that sense a powerful inhibitor for multiple different glycosidases. So glycosyrine is an effective novel multifunctional glycosidase inhibitor. Now, this is not only occurring in DC3000, it's a gene cluster that occurs in many different Pseudomonas syringae, and we think it's able to move by horizontal gene transfer because it's flanked by transposons and has a lower GC content. All these different Pseudomonas syringae species have been isolated from all these different crops. So you can imagine if they indeed make these inhibitors during infection, these inhibitors were very likely able to suppress the glycosidases of all these different plants. So all these different mechanisms that you could think about may occur during the infection of all these different plants. So basically that means there's a lot of glycobiology that is manipulated by plants, by this plant pathogen. So we have addressed why and how this BGAL1 is suppressed. But is this the only enzyme that is suppressed? Well, no. Daniele Sueldo took it a big step further by mixing 3 different probes that target different classes of hydrolases. And all of them are biotinylated, so what she did is she labeled, then pulled out the biotinylated proteins and did mass spectrometry. And in this way, she was able to monitor 171 active hydrolases in an aplastic fluid. And when she did that, comparing infected with non-infected material, she could see there were about 100 enzymes, hydrolase activities going up. Most of them are serine hydrolases, but also about 60 activities going down. And these are mostly cysteine proteases and glycosidases, and that includes BGAL1. So there's more, there's about another 50 or 60 enzymes more that are suppressed. So how do we go with this further? We take advantage of off-fold multimer. So off-fold multimer allows you to predict protein-protein interactions. So what Fiet Oma did in the lab is he took the 48 suppressed hydrolases that were detected in aplastic fluids, and also took 211 detected DC3000 proteins that we know are present during infection. So what we did is we basically look at all possible complexes between the plant hydrolases and putative inhibitors. So we screened 10,000 protein pairs in the computing cluster at Oxford University, and we get hits for every of those pairs. So what we see here is 504 protein pairs with a good interaction interface score. So there's a good complex between the two. We then manually looked at all of them, the 504, which is okay, that's 2 days of work, and found that actually about 30 interactions are very likely putative inhibitors interacting with hydrolases. So there are about 30, potentially 30 different secreted hydrolase inhibitors produced by by Pseudomonas shringi. And just to give you one example, this is a very nice hydrolase inhibitor. So the hydrolase is in gray, the active site is in red, and in blue you see the inhibitor from the bacteria. It's blue because it has a very high so-called PLDTP score. That means we are quite sure about the structure. And this is the electrostatic interaction. It's an absolutely perfect inhibitor and long interface along the substrate binding group. And this is an interesting enzyme. It is neofunctionalized. It's not a chitinase anymore. It does something else, but it is important in immunity, and we are investigating that. If you are interested in AFM screens, we just published some tips that you may want to know about when you want to do this. Okay, conclusions are that DC1000 suppresses dozens of hydrolase during infection. Bigol is one of them, and it is inhibited because it promotes the release of the flagellin elicitor. SPD5.2 is a subtilase that inactivates the epitope of flagellin. We have identified a novel immunosugar that manipulates host glycobiology beyond what it would need to do to suppress flagellin recognition. And we show that AFM can be used to predict inter-kingdom interactions between hydrolases and inhibitors. And with that, I'd like to thank the lab. This is when we are not wearing lab coats. And this is when supported by a couple of grants and collaborations. And I'd like to thank you for being here. And thank you for your attention.
Hola. Hola, chicas. Bueno, pues, gracias por venir al seminario. Creo que ahorita tienen la oportunidad de preguntar cosas, preguntas que puedan tener, dudas. O no nada más del seminario, entiendo que pueden preguntar de todo tipo de preguntas. Jóvenes investigadores, todo el mundo puede preguntar. Sí.
Thank you for your talk, Professor. I was curious about, you have phenotypes for the gene cluster that you say like A and B were essential while B, C, D were not. And looking at your slides, one of them was a phosphatase, and I guess you can get redundant functions from elsewhere. But you did not mention about the other ones. Do you have any idea if this kind of redundancy could?
Yeah, good question. Yeah, we think there's redundancy. So the transporter is from a larger family, and they are known to be promiscuous a little bit. So we think another transporter might do a good job as well. This a partial contribution, but not an essential contribution. The phosphatase, same thing. There are probably other phosphatases that can do this. So that's maybe why we don't see a strong phenotype. We do not know what the other component is doing. We have no idea. Yeah, but it works. Yeah.
¿Alguien más que quiera? ¿Alguna inquietud personal? Sí.
Looks like I'm on my own. People are a little shy to talk in English, so give them a chance. And about the origin of this, you show like this phylogenetic profile that you are showing the gene cluster, how it moves. But I was not clear, was it only on Pseudomonas or have you looked elsewhere? Where is it? I'm kind of curious. Working in the micro plant interface, it would be like some kind of tool to get this moving. I don't know if you're looking further into how this cluster is moving across all the bacteria.
No, yeah, good question. We do know, so we see, if you look at the phylogeny of the cluster, it's not congruent with the phylogeny of the bacteria. and that is one strong indication that the cluster has moved. We see that the cluster is flanked by transposons or fragments of transposons, and it's located next to a tRNA sequence, and that's also a hallmark for transposon insertion sites. It also has a lower GC content. Many Pseudomonas stringi have it. We tested one of them, that's a bean pathovar, which I haven't listed, but clearly that is able to make an inhibitor during infection of bean. So that's, I think, illustrates that this is very common. When looking at all the different bacterial species, other beyond Pseudomonas stringi, we know it's in Acidovorax, it is in Casiconia and in Erwinia. And they are known to be plant associated, not necessarily pathogenic, but plant associated. We don't see the cluster outside that group of bacteria.
Hi, thank you very much for your talk. And well, my question is more in general. These kind of responses, how specific are from the plant towards only bacteria, and what's like the first point for the plant to respond as that, and how general it is the response, for example, against fungi or something like that?
Yeah, so in general, plants respond very similarly to all kinds of pathogens. So bacteria, oomycetes, viruses. The immune response is universal. Yeah. So it's a long list of genes that are activated. But of course, the different, whether it has an effect. So, for example, a chitinase that is induced by upon infection with viruses and bacteria only really has an effect on fungi. So you would expect that fungi will have inhibitors, but all the other pathogens don't care about the chitinases. So that's what I would predict. That means there is specialization on the pathogen side, depending on whether they are sensitive for these hydrolytic enzymes.
So it's always like an exaggerated response from the plant?
Yeah, it is an over response. So plant immunity is relatively general. Yeah. So it is more how well the pathogen has adapted to a specific host. Mm-hmm. And that's why we don't have so many generalists. Most are very specialized pathogens. They figured out how to suppress immune responses in that plant species. Mm-hmm. And they specialized on it, and this is when they can cause disease.
Yes, thank you.
¿Alguien más que quiera participar? I'm curious about the computer power you need for this AlphaFold analysis between things from the two different organisms. A lot,
yeah, and I don't really know. It's a lot of energy that you use. I'm not so sure if the lights go out in when we start calculating structures, but we needed to use for 10,000. We used the computer facility for almost full time for about 2 weeks.
Full time, 2 weeks.
So it's substantial, but in Oxford and many other places, investments in better chips, especially GPUs, they really advance the speed. I can also recommend people using AlphaFold 3, the online application. You need 10 minutes to get a structure predicted.
One by one.
One by one.
Yeah.
But if you have a couple of students, you only need a Gmail account. Yeah. If you have 1,000 students, you can do 20,000 a day. But I know people that actually do it that way. With a couple of accounts, you can actually do screens. You can make some Google accounts by yourself as well.
Yeah. 10,000 Google accounts.
But I think an important thing to realize, something that we only realized after discussing with DeepMind, is that AlphaFold3 is trained on everything in the PDB database. That includes ions and it includes cofactors. If you don't add those in those screens, then the scores will be low. AlphaFold2, however, and the AlphaFold multimer is based on AlphaFold2. That is trained on the protein portion of the PDB database. So if you want to screen for protein-protein interaction, AlphaFold2 might still be better because it's difficult to really know which parameters you should add in AlphaFold3 to get the right complexes. Yeah.
Okay. I have another question because I think I didn't pick up before, but this inhibitor also inhibits proteases? Is that no?
The inhibitor of the glycoside?
Side.
No, we don't think so.
You don't think?
No.
Okay, I misunderstood that. Okay.
No.
Any other question? Yeah.
I was wondering if you can explain a little bit about the IPTM predictions. How do they work? Out of complete ignorance, how do you manage? You show this huge plot where you say like these interactions are like significant. I don't know.
Yeah.
Then
Yeah, we benchmark. It's a bit of a black box. The AlphaFold predicted complexes, the interaction between residues of both proteins, that's the distance and the confirmation how well they fit, that gives a combined score. And that is the IPTM, the Interface Predicted Template Modeling Score, the IPTM. We did notice, and many others as well, that some interactions are not real. AlphaFold cannot distinguish. For example, if you have a good interaction, you make a mutation and you know that that interaction does no longer work, AlphaFold can still predict exactly the same score. Yeah, so we need to go back and look at the interface at a different way to extract the relevant information to say this is a good or this is a bad complex. Yeah, it's very much a black box, to be honest. Yeah, empirical science. Yeah.
¿Alguien más? ¿Alguien? Me, another question. So one, sorry, that is just in this screen that you did with the multimers, that has nothing to do with the inhibitor. That's just proteins from the bacteria and proteins from the plant trying to find out pairs that interact. Yeah.
Yes, we are looking for inhibitors in that screen.
Inhibitors, but not of the type of the glycan.
No, I mean we have. I think we did. I think put in Beagle as well as a control. It did give us a hit, by the way.
But in this case, the inhibition would be from a protein from the bacteria to a protein of the plant.
Yes.
Yeah.
And the focus that we have, and I think it's an important trick. We know these enzymes are suppressed. We only took 48 that we know experimentally they are suppressed during infection. It could be through a small molecule or a protein or maybe another mechanism. Yeah, but let's assume if it is a protein, there are only 211 proteins that we could detect. So we thought, let's take those and see if we get complexes that make sense. Now, I did mention we get 500 plus, over 500 hits. It's way more than it's conceptually possible to be inhibitors. But if you look at the predicted structure, it's very easy to actually say, oh yeah, that's the hydrolase is like this, the active site is there, but the protein interacts there. That's not an inhibitor. It may be an allosteric regulator, but we ignored those. And then we found out that only 10% are of this type, where you have the inhibitor fitting nicely in the pocket, blocking the active site. Yeah, so that's 10% of the, well, less than 10% of the 500 that we had to look at.
Sí. Una aquí antes y después.
I don't know if I lost that part, but I was wondering about the release mechanism of this inhibitor. I mean, because you know all the pathway for the biosynthesis, but about the release mechanism.
Oh, well, it's yes, from the bacteria. Yeah, no idea. We have a transport in the cluster, but it's partially required for inhibitor production, but we don't know if that's because it is involved in the import of molecules, I'm not sure, or secretion of molecules. We don't really know. So no, we don't know. I guess there is a transporter involved. It's not something that would just diffuse out of across a membrane.
And somehow it is included in the transporter in this cluster, in this genetic cluster.
Yeah, the last gene in the cluster is a transporter.
Okay.
Yeah, but that was partially required.
And just another question about if there is any mechanism similar in roots or something about the pathogen, like send inhibitors to avoid enzymes activity or something.
Very likely. We haven't studied that, but I think that's very likely. I think the secreted proteome of the plant, there will be differences, but there will also be a lot of similarities. And that means there will be also inhibitors from adapted pathogens to suppress those. Yeah.
Just one question. How sensitive are these enzymes to pH? I mean, because there are also many proton pumps that are involved in this response.
Yeah, well, it's a very good question. I can't answer that because we didn't test it, but it is an important component. We know for other interactions, some inhibitors only work at low pH.
Yes,
for example. Yeah. But for this one, we haven't tested. We do know, however, that the inhibitor is unstable. It is not sure what chemically happens with them, but they are inactivated at a higher basic pH, above 8. But that's maybe not physiological.
Yeah, okay.
Más. Pues sí, por favor. Pues muchas gracias, chicos. Espero que les haya gustado, ¿no? Obviamente se tocaron puntos que a lo mejor ustedes, si son de los primeros semestres, todavía les cuesta trabajo entender. Muchos detalles, ¿no? Yo le había pedido que fuera un poco más general, pero le gustó porque creo que les acaban de aceptar este artículo en Science, entonces está, claro, es toda una vía que acaban de descubrir y quiere como mostrar lo que se puede hacer, ¿no? Pero obviamente en este tema de interacción planta con microorganismos, se pueden ver muchos puntos de vista, desde el punto de vista de la planta, desde el punto de vista de los microorganismos, que en este caso pues él nos mostró de bacterias, pero obviamente siempre hay una guerra así le llaman una guerra armamentista, porque la planta se defiende y el microorganismo evoluciona para poder seguir infectando, y entonces es así, ¿no? Van evolucionando, coevolucionando los dos organismos. Entonces hay mucho, es un tema muy activo, muy bonito. Además, nosotros estamos ahí trabajando también con este tipo de cosas en el instituto, y por eso tenemos una colaboración con el profesor. Y bueno, pues ojalá les haya gustado, que hayan aprendido, que sepan que pues la biología es complicada, pero bonita, si la estudiamos bien. Thank you very much, dear Renier. We have been discussing about all the different things that you can just learn by studying interaction between two different organisms, very different, but the coevolution among them it goes all the way. It has occurred since the beginning, and yeah, it is still happening, and it's exciting to find out all these details of how the organisms interact and live together. And thank you very much for coming here and talk to us today.
Bueno, para la Secretaría de Comunicación de la Facultad de Ciencias ha sido un honor que la doctora Claudia Martínez, pues nos haya en realidad ofrecido la participación del doctor esta mañana y pues en realidad aceptamos gustosos la colaboración del Instituto de Biotecnología y pues ahora solo invitarles a que si quieren acercarse a este lado del auditorio con la idea de que podamos tomar unas fotografías para que podamos tenerlo como registro de este momento en el que ustedes están asistiendo acá. Les agradezco. Por favor, si se acercan a este lado del auditorio para que el doctor baje y se coloque, tomamos la fotografía. Acercarse, chicos, al centro. Acá, si quieren, por favor.